Good afternoon, everyone, and welcome to the pitching session on call 7, topic 3, on clinical validation of biomarkers for diagnosis, monitoring disease progression, and treatment response. So before we start the session, I would like to remind you that the session is recorded as well as the presentation, and we will be publishing everything on the IHI website and on the B2Match platform by the end of today. I would also like to say to the pitchers to please strictly follow the 4-minute timeline for the pitching. And you, as attendee, if you are interested in any of the presentation given by the pitchers, please, I recommend you to contact them on the B2Match platform. So on the B2Match platform, you can easily find them and contact them, message them, or setting up a meeting as well. So now we will start the presentation with our first speakers, Cristina. Cristina, the floor is yours.
Thank you. So my name is Cristina COZZINI. I'm working for GE Healthcare, so I'm speaking on behalf of the GE Healthcare team. And I'm also happy to announce that for this proposal, we are partnering with the Policlinico Gemelli in Rome, with Professor Sala and Boldrini taking the clinical lead for this project. What we plan to propose is biomarkers for AI-enabled patient-specific treatment for cancer in radiotherapy. Next slide, please. So radiation therapy is received by more than half of all cancer patients during their treatment path, and often is combined with other treatment options like chemotherapy and immunotherapy. And there have been tremendous advances in recent years into the radiation therapy field. And the field is moving towards tailoring more and more the dose delivery to the tumor and minimizing the dose to the healthy organs. And since all these treatment options are increasing, there start to arise new questions and new unmet clinical need to select patient-specific treatments and also to optimize the treatment response monitoring. And we need to answer to questions like, how do we determine which therapy or combination of therapies are optimal for the specific patient, and how can we balance the need of treatment with the toxicity risks, or can immunotherapy boost the therapeutic value of radiation therapy? Next slide, please. So what we plan to propose is to develop a digital platform and a set of PET and MRI imaging biomarkers that, together with non-imaging biomarkers, will enable AI analytics for cancer treatment response prediction and monitoring. The objective is to minimize the toxicity risks, assess radio and drug resistance, and predict treatment response for a better patient stratification and the optimized patient-specific outcomes. So what you see here in the schematic is a simplified path of the patients throughout the treatment. The diagnostic part is grayed out, so we are not looking at the diagnostic, but everything that comes afterwards. So in the response prediction, treatment selection, toxicity and resistance assessment, radio and drug sensitivity, patient stratification, and monitoring. What we bring in as GE Healthcare is expertise in imaging biomarkers, particularly with carbon-13 as a biomarker, multiparametric MRI, and then also the PET diagnostic aspect, so PET tracers, and of course the combination of PET and MRI imaging. What we also bring in is a data platform. For this, we are also leveraging the work that is being done also with the support of IHI for a project called Predictom that is developing a screening platform for neurodegenerative diseases. So we intend to leverage the work that is being done for that project and to expand it also for the biomarkers for cancer in radiation therapy. Next slide, please. So what we are building upon, as mentioned, the Predictom project, but also other EU-funded project like the AIT Health, where with the consortium called the DeepMR Only Radiation Therapy, we have developed tools in order to implement the MRI imaging into the radiation therapy workflow. For this, we are working with centers like the Erasmus Medical Center, the Newcastle University, and the Trust. And now we are partnering with the Policlinico Gemelli, which has a very comprehensive cancer center, which is serving more than 50,000 cancer patients. And they have a very wide portfolio of research projects and scientific publications. Besides, we are also having a discussion with other preliminary partners. We have been having also some preliminary discussions with pharma like AstraZeneca in the context of the carbon-13 biomarkers, and we'll expand those discussions in the coming days. What we are still looking for in terms of expertise is our companies which bring in-kind contribution with expertise in non-imaging biomarkers in the context of cancer in radiotherapy. As far AI tools are concerned, we do have expertise in feature extraction from images, and we would appreciate support into the response prediction and treatment monitoring aspect. So this is the very general overview of what we plan to propose for this IHI call, and I thank you for your attention.
Thank you very much, Cristina, for your presentation. And now our next speaker is Tobias.
Good. Hello, everyone. My name is Tobias HECKEL. I speak and present to you our project BREETHE on behalf of my colleagues from Siemens Healthineers. BREETHE stands for Biomarker-aided Refinements in the Assessment and Treatment of Lung Cancer for Health Enhancement. Next slide. What are the problems with lung cancer? Lung cancer is the leading cause of cancer death with about 2 million new cases and about 2 million deaths every year worldwide. The lung is feeling no pain, which is why lung cancer is often detected too late. Lung cancer survival rates can be increased through screening as well as by precision medicines. Biomarkers are crucial to identify patients who will benefit from therapies, and they have tremendous potential to improve early detection of lung cancer. Therefore, we urge to verify clinically applicable biomarkers and to combine image-based, digital and blood-based biomarkers in a multimodal fashion to improve patient outcomes along the entire patient journey. Next slide, please. Along the patient journey, we want to verify multimodal biomarkers with the early detection of cancer, as well as for risk stratification. Apply new technologies for the collection, purification, and measurement of biomarkers. Verify biomarkers to improve treatment selection and to improve access to personalized medicine. We want to provide a digital platform enabling the analysis and interpretation of multimodal data, and eventually develop a regulatory strategy, including the possibility to include liquid biopsy into reimbursement pathways. All these activities will generate better outcomes in lung cancer through the integrated use of multimodal biomarkers and ultimately contribute to the reduction of mortality. Next slide, please. I want to show you an example where we want to achieve our objectives. With clinical partners, we want to advance clinical studies where we can validate the combination of liquid biopsy with imaging biomarker for early detection of cancer in high-risk people, but also in people outside of the screening criteria. The goal is to reduce the false positive rate, especially at baseline CT scans, to refine lung cancer risk assessment, and to better stratify positive CT scans into low or high-risk categories. Moreover, we want to include liquid biopsy and imaging biomarker to predict therapy response and who will benefit from treatment escalation or de-escalation. All these measures will lead to earlier detection of cancer, faster diagnosis, and less interventions on benign findings. Next slide, please. To clinically validate biomarkers, we have identified 10 clinical partners expressing a very high interest working with us, and at least 2 SMEs with more to come via the IHI marketplace. We at Siemens Healthineers offer expertise in imaging and blood-based biomarkers in a clinical setting. With our TeamPlay digital health platform, we offer access to cloud computing systems for data sharing, as well as AI solutions like AI Red Companion to interpret medical images. Moreover, we have developed AI-based prototypes currently in clinical trials using image-based biomarker for radiotherapy personalization. Finally, we can offer one workspace for integrated diagnostics with our software solutions like SyncoCarbon. Next slide, please. We are looking now for partners that complement Siemens Healthineers' diagnostics, imaging, and radiotherapy capabilities, like pharmaceutical partners supporting biomarkers for precision medicine, technology development and biotech partner, providing novel technologies for sample preparation and sensing, and additional candidate biomarkers as well for validation. We're looking for clinical partners and have found already 10 interesting candidates who can give us access to samples and who can generate with us clinical evidence. We are interested in regulatory partner interactions to prepare for the approval process of biomarker and explore the path to reimbursement. And we are open to patient representations to support access to more biomarker in lung cancer. Thank you for your attention.
Thank you very much, Tobias, for your presentation. And now the floor is to Ralf.
Yeah, thank you. Good afternoon. So my name is Ralf HOFFMANN, and working for Philips, and you can see here my contact details, and I'm presenting the BRECISE project, which is an acronym for Biomarker Research and Evaluation for Clinical Implementation and Supporting System Enhancement. Next slide, please. So from our perspective, the challenges, especially in the oncology field, is that we have a limited number of prognostic and predictive biomarkers that are subject to rigorous clinical testing and therefore hindering the progress in precision medicine. And furthermore, if we look at access to affordable platforms, especially in the field of NGS, next generation sequencing, but also modality, we have a lack of these platforms here for clinical use with fast turnaround times to get them access in an effective way to the clinician. So therefore, we suggest a project where we don't necessarily bring forward a specific biomarker or a specific disease, but we would suggest to create a framework that supports oncology-driven biomarker validation and also scaling, not only for now with a few use cases that we suggest, but also that a framework that can be used in the future for this purpose. We suggest to enhance one of the objectives associated biomarker technologies for NGS in particular, establish a collaborative data sharing platform for scaling of clinical biomarkers, provide a regulatory and market acceptance strategy, and develop dissemination strategies and also educational resources. So the impact that we expect is that on one hand, the availability of some clinically validated NGS biomarkers to support the healthcare professional in treatment decision, improving outcome and patient satisfaction, but also validated biomarker technologies to enable future research and therefore increasing competitiveness of European health industries. Next slide, please. So these are the main activities. Don't look at this as like a work package structure, although it may look like this, but the important part is the box on the bottom with these biomarker use cases, where we already have some partners and some use cases that we're discussing in more detail, for instance, in prostate cancer and bladder cancer, but they are not fully defined yet. But we want to use these use cases along with biomarker NGS technology companies to create this framework that is complemented by regulatory integrated data platform AI technologies in order to, as I said, provide a framework for future biomarker validation to scale this whole field, which we believe is strongly important. Next slide, please. So the expertise, we already have something like 10 plus partners that are very interested to work with us. We provide ourselves knowledge in biomarkers on the NGS side in the oncology domain for multiple use cases with a certain level of validation. We have a few clinical partners here on board that are interested to work with us, and we are definitely committed to bring also ICOP and ICA into the project for this purpose. Next slide. What we are still looking for is in particular pharma companies that are interested in supporting the clinical trials that we aim to do, especially in prostate and bladder cancer, or biotech companies with the same purpose. Biobanks and data registries for access to biosamples and clinical data, as well as regulatory expertise in this field. Thank you very much for your attention.
Thank you very much for your presentation, Ralf. So now our next speaker is François-Clément.
Hello, everyone. So my name is François-Clément BIDARD. So I'm a medical oncologist from academia, and I'm so coordinating breast cancer research at Institut Curie, which is the largest center in France for breast cancer and also in the EU. We also have been awarded and recognized as the National Institute for Women's Cancer in France by the French government. So next slide, please. So excellent environment to do research, but also to do translational research. And we have also an industry-friendly setting with a lot of ongoing collaborations. So we are used to set up this kind of private-public consortium. Next. So we are building this consortium on our experience past experience in demonstrating the clinical utility of biomarkers, and just I want to highlight two of our very recent achievements. One is the first and only trial that has shown the clinical utility, so which means improvement in patient survival in a randomized phase 3 trial for circulating tumor cell count, and with very good publication. And also my team and I set up the PADA-1 trial, which is also the first and only trial to date to demonstrate the clinical utility of ctDNA monitoring in metastatic breast cancer patients. So it's not only like pure academic exercise, but we also delivered value to our private partners. The CTC company is now applying for reimbursement in Europe, and PADA-1 we have industries that are now running registrational trial based on our design. Next slide. So what about your consortium? So the idea is to build around the patient pathway in metastatic breast cancer. We have now several antibody drug conjugate, NR2, Trodelvy, and new others that are coming in the near future. And the idea is to launch what we could consider as a biomarker campaign. I'm sorry for the typo here. So before, during, after. I'm especially interested in looking at functional imaging, which is PET-CT with new radio tracer combined with AI analysis. It would come with other biomarkers, namely tumor biopsy, multiplex digital pathology, which is, I think, very important to choose between different antibody drug conjugate, and AI analysis plus minus spatial transcriptomics, depending on our partner. Of course, liquid biopsy markers, but again, so the idea is more to go first on new radio tracer. Primary objective would be to validate one of the biomarker, like really to push a biomarker toward, let's say, pre-registrational step, and also to explore other biomarker. We could also consider, depending on the biomarker technology readiness level, some more complex statistical approach. And here on the scheme, I put an antibody or several antibody drug conjugates, but we have also, depending on our partner, especially pharma partners, could be also an AO agents and IO related radio tracer, or we could also think about something about vectorized radiation therapy with therapeutic radionuclide. Next one. So what we are looking for here is first radio tracer. Of course, we started to discuss with companies, but still interested in getting new clinical grade radio tracer related to ADC targets. So relevant for the breast cancer field, so HER2, TROP2, ADC, digital pathology, spatial transcriptomics, ctDNA, CTC. We already secured everything that is needed to run the consortium, and I would be very glad if you contact me for to know more about our trial. Thank you.
Thank you very much, François-Clément, for your presentation. And now the next speaker is Daniela.
Thank you very much for the opportunity to share the overall concept of DOMINOES with you today. I'm Daniela Cirillo from the San Raffaele Scientific Institute in Milano, Italy, in partnership with several partners of previous IMI project and the EDCTP project on diagnostics. DOMINOES is about moving bacterial diseases diagnosis and treatment toward a precision medicine approach. We strongly believe that an early partnership between diagnostic manufacturer, drug manufacturer, and academia, such the one we are proposing here, is needed to achieve this goal. Next. Okay. Antimicrobial resistance is one of the top recognized public health priorities. By ECDC report, Southern European countries are registering alarming rates of bacterial pathogens resistant to first-line therapy, both in nosocomial environment and in communities. It is estimated that in 2050, up to 10 million people will die for an AMR pathogen, matching the death toll that we are paying for cancer. Bacterial-related biomarkers detection can provide an answer and impact this negative trend. Bacteria-specific biomarkers able to provide species detection and markers of drug resistance can guide personalized treatment, highly increasing rate of success and minimizing drug resistance generation and pathogens transmission. Data show that the line in the use of proper diagnostics and the blind use of empiric treatment in settings at high AMR rate are both contributing to selection and an increased transmission of those resistant pathogens. Introduction of new drugs without diagnostics to facilitate their proper use in selecting the patient that will benefit from that specific drug are not impacting drug resistance in the long run. Let's consider the story of the new therapy for drug-resistant TB. New drugs were introduced without development of proper diagnostics tools and biomarkers to detect the drug resistance, not only at the patient's level, but also at the surveillance level. And that led to a poorly controlled increase of resistance and failure of therapy, jeopardizing the role of those drugs for the future. Next. Our work plan focused on reverting this process by discovery optimization and clinical validation of pathogens-based biomarker using the highly innovative technologies that are available to us today that are able to reduce to a minimum time the diagnostic delay. Our target are biomarkers for bacteria, mycobacteria, mostly M. tuberculosis, fungi that are able to address diagnosis, drug resistance, and treatment monitoring. We will validate them, in particular those that are close to commercialization, designing proper trial to collect evidences to move them through regulatory pathways. Next. Who are we? We are a consortium strong in AMR bacterial pathogens detection with high level of capacity to collaborate with industries that are already on board. Our partners have worked together in previous EU and non-EU funded drug and diagnostic trials. We have largely contributed to WHO diagnostic and drug policy in the past with our results. One of our partners is a leader of diagnostic trial in low to middle income countries. We do have regulatory expert and patients organization on board. Next, what are we looking for? We are looking for partners with innovative technology we may not be aware of right now that are able to contribute scientifically and financially. We will guarantee a company-friendly and oriented collaborative approach to overcome any competition issues. Thank you very much.
Thank you very much, Daniela, for your presentation. And now the next speaker is Milena.
So good afternoon, everybody. I would like to extend a very warm thank you for the ability to present the project on behalf of our consortium, which deals with the clinical utility and implementation of circulating tumor DNA across the patient journey. I think the last part of the title cannot be highlighted enough, and as we've seen in the previous pitches, we really need to work on advancing the patient journey across the story. Next slide, please. The challenges and objectives that we as a consortium have identified have been dealing with ctDNA biomarker clinical applicability. It has been demonstrated and we do know who, how, and when to treat. However, the clinically applicability of ctDNA testing is still not at a level that we are confident with. We would like to focus on two key points: the utility and the implementation in clinical practice and the regulatory pathway. The main objective of this project would be to improve the care for patients with cancer by bringing ctDNA molecular diagnostics to clinical implementation. What we expect as outcomes and impact is to improve ctDNA-guided precision medicine, specifically to be able to better the selection of patients, to detect earlier detection of recurrence, to precisely monitor the therapy response, and to improve the quality of life of our patients with cancer. Also, we need to focus on the EU regulatory framework, as we need to qualify and focus on reimbursement and implementation. Next slide, please. The main activities that we will be focusing on within this project are the utility and implementation of the circulating tumor DNA across the patient journey. We will focus primarily on colon and rectal cancer as two biological entities, and we will monitor the prognosis and surveillance, treatment selection and prediction, and treatment response and monitoring, both in the metastatic and non-metastatic settings. This will, of course, be very useful for other types of cancer, as once we have proven that the circulatory tumor DNA can be useful across the patient journey, it will be applicable from colorectal cancer to other cancer types, thus increasing the overall impact in the coming years. We will also focus on fair data stewardship and health technology assessment. In the implementation part, we will focus on 3 A's: availability, affordability, and accessibility. We will involve and organize a variety of stakeholders which are already in place at the national and at the EU level. Next slide, please. The resources and expertise that we offer are within 3 countries: the Netherlands, Denmark, and Serbia. Our expertise is within colon cancer and rectal cancer observational and interventional ctDNA biomarker studies, which are already en route, and we have high expertise within these cancer types and other types that we wish to implement it further on. We offer fair data stewardship expertise, as well as health technology assessment expertise, and a professional company with program management and communication to ensure a soft and easy flow of the project. Our resources are also availability of clinical study data and samples, which will be available after the project as well. We do have the infrastructure to run multi-center and multinational prospective clinical validation studies. Next slide, please. What we are searching from within the expertise is to find ctDNA biomarker companies to help us clinically validate ctDNA assays and pave the path to its regulatory qualification and reimbursement in a real-world setting. We would also like to invite pharmaceutical companies, imaging and AI companies, as well as regulators involved in biomarker test regulatory qualification, reimbursement, and implementation. If I might add, we are a consortium of researchers with high integrity and a genuine interest in the patient journey. Thus, we invite you to contact us after the pitch, and we hope that this project, whether successful or not, will stem off new and useful collaborations for our patients in the future. Thank you very much.
Thank you very much for your presentation, Milena. Now the floor is to Hendrik.
Hello, thank you for the opportunity to present to you our ideas about biomarkers. Why are we here? Next slide, please. Sorry. So we are here because we have been active for several years in the Quantitative Imaging Biomarkers Alliance from the RSNA, and we produced a profile describing how to do DC-EMOI correctly or most quantitatively. We reached with the profile, which is a kind of manual, reached consensus stage, which means that lots of experts were giving us input about what to do and what not to do. And now we can provide calibration and standardization workflows for DC-EMOI. We still continue to do monthly meetings every second Monday of the month at 6 p.m. You're all welcome to join us if you're interested in dynamic contrast MRI. Next slide, please. Who are we at the moment? There are several people involved. Most actively is University Center in Amsterdam and Erasmus MC in Rotterdam, and Fraunhofer Mewis, which is a research institute located in Germany, where I'm sitting. Next slide, please. Dynamic contrast enhancer is quite old. It's been used, but it's always been promising. But well, so just for those of you who don't know it already, DCMR means that you measure the contrast agent you inject into the patient and look how the time course goes over time, and this gives you information about the microvasculature or perfusion of the tissue, and this can be helpful to monitor cancer and inflammatory diseases. Next slide, please. The problem is that DCMR, injecting contrast agent, is widely done. It's almost, especially in oncology, is almost every time it's being administered. What's missing is the quantitation, so the comparison of values afterwards. And therefore, we'd like to standardize the acquisition protocol and hardware, because there are several vendors for MRI systems. And to cope with multi-center trials, you have to get those all into one comparable setting, and we also want to standardize the analysis methods, the software afterwards, and we have methods to do this. Our goal with this is to have a widely accepted biomarker to predict outcome, to assess treatment efficiency, and also to adjust therapies if needed. We especially the radiologists in our team, think that most promising at the moment are bone and soft tissue tumors and also rheumatoid arthritis. And next slide, please. So, and for this, we'd like to set up a standardized quantitative workflow for doing DCMRI. And most importantly, make this clinically implementable, because it's always a lot of things to be done there, and to make it possible in every clinic is a challenge, but we are prepared to help with this. And our main goal is to motivate and aid in multi-center clinical trials, which will make DCMRI an established biomarker at some point. Next slide, please. So what we can do, so we have clinical data and expertise from multiple hospitals. We have experience with multiple vendors, and also with the requirements for sequence implementation, and also how to calibrate sequences and the software being used afterwards. So we have some software available. One is from our house, has motion correction, can calibrate T1 and B1 maps, and give you the give you parameters which are repeatable. Next slide. And what we are looking for, pharmaceutical companies in these kind of these topics with cancer and rheumatoid arthritis, or manufacturers of contrast agents might also be helpful. And clinics who are working in this field. Yeah. So thank you. 4 minutes is quite short, but thank you very much for your attention.
Thank you very much, Hendrik. Indeed, 4 minutes is really goes really fast. So our next speaker now is Karim. We cannot hear you, Karim.
Sorry, I was on mute. Yes, hello everyone. My name is Karim LEKADIR. I'm from the University of Barcelona, and I will present these slides on behalf of the AI for Health Imaging Network. Next slide, please. So I would like to focus on imaging biomarkers, in particular AI-enabled imaging biomarkers, which are promising based on the literature and the recent studies, such as radiomics, radiogenomics, deep learning-based biomarkers, and so on. Next slide, please. But the challenge is that there's a lack of validation evidence regarding their efficacy, safety, trustworthiness, reliability, reproducibility, and clinical deployment has been very limited. Next slide, please. So what we'd like to do is bridge this gap by offering a comprehensive, holistic validation platform that will be linked to European data across all EU regions, define standardized validation criteria, metrics, and method for imaging biomarkers, and implement a number of use cases, maybe 3 or 4, that address clinical and industrial needs. Next slide, please. So what we offer is expertise and resources that were accumulated over the years through the AI for Health Imaging Networks that comprises a wide range of EU projects in the field of cancer imaging. In particular, there's also the Cancer Image Europe, which is a very big project with 75 partners linking different data repositories in Europe. Next slide, please. So we have really a wide range of institution expertise, clinical sites. We have expertise in AI data, clinical validation, and ethical and legal issues as well, health economics. Next slide, please. So we have the federated data repository I mentioned that will have over 100,000 patients and also millions of imaging, non-imaging data, and we can cover a number of clinical domains. Next, please. This is a number of tools and solutions that we've developed over the years that we could also build upon in this project to build this comprehensive validation platform. Next slide, please. So the main work will be to develop this validation platform that will comprise a data catalog to search for image data that will be relevant for a particular use case. There will be a federated data access platform to access the data, in silico validation, prospective validation, also continuous validation and monitoring. This is important because these biomarkers vary over time, depending on the equipment and the protocols, and of course, regulatory approval. Next. So we can cover also a number of clinical use cases: breast, colorectal, prostate, liver, lung, brain, and cardiac imaging data. We will select 3 or 4 use cases depending on the need of the pharmaceutical companies and clinical sites and so on. Next slide, please. So please contact me if you're interested. So we're looking for technology companies, pharma companies. We're also looking for clinical side that will help us define the use cases. So we're quite flexible at this stage within the 7 domains that I mentioned. Would like also partners that will help us develop the platform, define the needs, lead the clinical validations as well, and then define exploitation routes. So these biomarkers will be validated, but hopefully also as a next step deployed in clinical trials and also in clinical practice. So I look forward to hearing from you and putting you in touch with the other partners of the AI for Health Imaging Network. Thank you.
Thank you very much, Karim, for your presentation. And now the floor is to Michael.
Hello, I'm Michael Levine from Imperial College London, and I'm pitching on behalf of the EU-funded Diamonds Consortium on our project to bring biomarkers for infectious and inflammatory disease into clinical use. Next slide, please. Infectious diseases are amongst the most important problems and are in need of urgent improvement in diagnosis, particularly to reduce indiscriminate antibiotic use and to reduce antimicrobial resistance. You will all be aware that many thousands of biomarkers are published in the literature, yet very few of them lead to clinical use. And our project aims to bring this gap and to accelerate the development and introduction into clinical use of biomarkers for infectious and inflammatory diseases. And we hope within the course of the project to progress multiple host response RNA or protein biomarkers into regulatory approval and clinical use. Next slide, please. The way that we will do this builds on 20 years of large-scale EU funding through successive multi-country grants. Our current Diamonds Consortium has a biobank of over 20,000 patients with infectious and inflammatory diseases, both children and adults, with detailed clinical data on our patients and with biobanked plasma, serum, RNA, and DNA, and with extensive RNA sequencing, proteomic, and pathogen data on the subjects in the study. We have over many years of successive grants developed an experienced clinical network at 27 hospitals in 11 different EU countries, as well as partners in Africa and Asia. And we have published in high impact journals and patented a number of RNA and protein signatures for common and important diseases. Our pipeline can be entered at any point. A candidate biomarker, either one of our own or from a partner, can undergo in silico validation in our RNA and protein data. Promising biomarkers can be evaluated on stored samples and then progress to near patient technologies in cross-platform translation using stored samples. Prototypes can be evaluated first on biobanked samples, then on independent samples, and those that look promising can progress to clinical use and clinical evaluation by our clinical network, which can also undertake cost effectiveness, user acceptability, and stakeholder acceptability evaluation, all important for regulatory approval and introduction into clinical use. Next slide, please. Our consortium has extensive experience through successive large EU grants in all phases of the biomarker progression pipeline, from clinical recruitment, RNA and proteomic analysis, bioinformatic and biomarker discovery, test platform evaluation, and cost effectiveness and utility. So we have published and patented numerous biomarkers for bacterial and viral infections, TB, malaria, and other inflammatory diseases. Next slide, please. We seek partners who can join us, particularly companies with candidate test platforms or candidate biomarkers or both, who wish to see them progress to clinical evaluation and want to work collaboratively with us using our biobank and our published and patented signatures and our clinical network. We need partners who can make a significant in-kind contribution and willing to work in a multidisciplinary group. And we also seek regulators, foundations, and advocacy group to join our consortium. I thank you for listening and would be happy to make contact with potential partners interested in our work offline. Thank you.
Thank you very much, Michael, for your presentation. And now our next speaker is Carl.
Hi, everyone. I'm Carles from Serbia, a health research institute located in Alicante, Spain, and I'm here to present you the proposal entitled Integrative Analysis of Biomarkers to Improve Monitoring of Neurological and Heart Diseases. Next slide, please. The challenge that we address is the personalized monitoring of disease progression. In this context, the loss of key biomarkers in clinical routine delay the timely administration of therapeutic intervention. For this reason, we aim to validate novel clinical biomarkers that could facilitate the early detection of disease worsening and help clinical decision-making in neurological and/or cardiovascular disease. How do we do that? Using a comprehensive, integrative, and technological approach to precisely identify and monitor crucial molecular and biomechanical biomarkers. Here, I present you some potential results and expected impact. For instance, result 1, AI-based risk assessment model for early identification of disease worsening. As for the expected impact, impact 1, our proposal will mitigate economic and health burden because the proactive identification of disease progression. Next slide, please. Now let's move to the main activities we propose. We could lead some of this one, the recruitment of patients, the validation and assessment of biomarkers, mainly based on circulating nucleic acids in body fluids and biomechanical parameters, the signal and biomarkers information treatment to further AI application, the AI-based feature discovery related to illness worsening, and the development of prognosis model. Next slide, please. Regarding the expertise and resource offer, we are an alliance of 3 Spanish institutions. Isabel, as a health research manager, as a health research institute linked to a public hospital, could bring and facilitate access to patient cohort and bring expertise in assessing fluid molecular biomarkers. UMH is a public university and brings expertise in the analysis of physiological and biomechanical signals. And ITI is a tech organization specialized in AI and ML techniques to develop predictive models. Here, I would like to add some information that is not included in the slide. Firstly, we could bring on a small and medium enterprise expert in the design and development of electronic products, advanced sensor, and wireless technology. Secondly, we are in talks with a pharma company, and hopefully in the following weeks, we could confirm its participation. And lastly, we have a good relationship with the policymaker, the Health Ministry of Valencia region, and if needed, we could also reach it. Next slide, please. So what we are looking for, basically we are looking to join a consortium on either of the following topics, topic 1, sorry, there is a mistake, and call 7, topic 3. And if needed, we could lead a Spanish subconsortium in the full proposal. And basically that means that we are looking for clinical partners to bring additional cohorts into these diseases that we mentioned before. Also, we are looking for research centers with expertise in the biomarkers presented before, and pharma industries and organizations with expertise in regulatory affairs. That's it. Thank you so much. Have a nice day.
Thank you very much, Carles, for your presentation. Now we have Lucia pitching on behalf of Donato.
Thanks a lot. Good afternoon, everybody. This is to outline how and why the European Pediatrics Translational Research Infrastructure could contribute in an HI research proposal dealing with biomarkers. Next, please. So first of all, why involving children in a proposal dealing with diseases prevention and treatment monitoring? This is because children might show unique vulnerabilities and diseases since early infancy, and you can see here a non-exhaustive list, and moreover, the availability of biomarkers could contribute to the faster characterization of drug efficacy and safety in the pediatric population. But on the other side, it should be considered that biomarkers identification and integration in pediatrics might be challenging due to methodological, logistical, scientific, and economic reasons. Next, please. So how EPTRI could contribute to these gaps? Firstly, a few words about it. EPTRI is a pan-European initiative acting as an enabler and delivering research services to boost pediatric research ecosystem. It acts as a distributed research infrastructure and is organized with central hubs and several spokes represented by a series of research units. As biomarkers is one of the EPTRI scientific domain, we would like to actively collaborate with consortia on the implementation of research activities to demonstrate the value of biomarkers as tool for prognostic purposes and for prediction of drug efficacy and safety. Next, please. So EPTRI contribution can count also on collaboration from research units working in other 4 fields of interest in pediatrics that are medicines discovery, developmental pharmacology, pediatric medicines drug formulation, and medical devices. Next, please. So to conclude, some more details on the thematic research platform on pediatrics biomarkers and biosamples that is composed by more than 30 research units from 14 European and non-European countries, and it spans across different therapeutics areas. So by matching the themes of the call with the services that EPTRI could deliver, 4 main areas of activities have been identified, and these refer to scientific and methodological aspects, networking, bioinformatics and data curation, and last but not least, regulatory and writing. So you can have here a list on potential activities that EPTRI could contribute and or lead in a collaborative project from the identification, characterization, and validation of biomarkers, access to deposit of biological samples, bioinformatics, large-scale screening studies in collaboration with pediatric clinical centers, use of biomarkers together with PK and PD modeling and simulation to support study design optimization and dosing algorithms, digital technologies and evaluation of biomarkers using advanced statistical and computational tool, delivery of novel point-of-care testing, use of biomarkers for diagnostic and prognostic purposes as basis for the prediction of treatment response, also in the framework of pediatric clinical trials. Systematic review of clinically relevant differences between adult and pediatric conditions, monitoring changes of markers by using wearable devices, and as last item, regulatory qualification process of pediatric biomarkers, regulatory compliant data sharing and material transfer agreement drafting, and collaboration with patients association and young person advisory group for having a tailored training and empowerment approach starting from early childhood. And next, just to thank you and to leave here a list of additional reference in case this could be of interest.
Thank you very much, Lucia, for your presentation. Now we will have Somik on the floor. The floor is yours, Somik.
Thank you very much. Hi, my name is Somik CHAKRAVARTY, and I'm a fundraising manager and a scientific grant writer at the Institute of Biological and Medical Imaging, IBMI, at Helmholtz, Munich, which is led by Professor Vasilis Diakristos. And I would like to present to you our expertise in imaging biomarkers and why you should consider us for your consortium. Next slide, please. Our expertise lies in developing groundbreaking technologies for biomedical imaging and sensing, such as fluorescence molecular imaging, and more importantly, the non-invasive techniques, optoacoustic techniques like MSOT and RSOM. Optoacoustic technology operates on the principle of laser light going inside the screen, and ultrasound is detected through the reflected waves, providing high-resolution images with excellent contrast. This allows for visualization of various tissue types and detection of specific molecules. The optoacoustic technology can resolve label-free novel disease biomarkers like microvasculature morphology and capillary endothelial function features, among many others which are listed here. And these are correlated with early detection and progression of diseases such as diabetes, cardiovascular diseases, psoriasis, melanoma, and many more. Next slide, please. We are the world-leading optoacoustic imaging research group. Our focus on clinically translating our technologies has resulted in numerous publications and prestigious awards. We have an extensive experience leading and collaborating in EU projects, which has resulted in several industrial collaboration and foundation of many spin-offs. Next slide, please. So how can we contribute to IHI Call 7 Topic 3, more specifically to your consortium? So building on our experience with optoacoustic and FMI imaging modalities, we offer established technology and methods for developing novel disease biomarkers for precise detection and patient-specific progression and treatment of cardiometabolic, skin diseases, and specific cancers such as breast cancer. We also do validation of biomarkers through standardized performance, repeatability, and reproducibility studies, including clinical validation studies. We have in-house expertise in developing and applying AI tools to extract and correlate biomarkers with disease detection and progression. Utilization of our field expertise in developing SOPs for biomarker detection and analysis. And lastly, we have a team of renowned scientists with an extensive network of clinical sites for preclinical and clinical studies. Next slide, please. For more details, visit our website and IHI Marketplace page. Contact us via email. I am Somik CHAKRAVARTY from IBMI Helmholtz Munich. Thank you for your attention. If you're interested in exploring collaboration or have specific inquiries, please feel free to reach out to us.
Thank you very much, Somik, for your presentation. Now our next speaker is William.
Thank you very much. My name is William Leenders. Indeed, I'm a Chief Scientific Officer of Predict Diagnostics company in the Netherlands. Next slide, please. As we all know, there's a great lack in oncology in tests for early diagnosis of cancer, which will greatly improve early treatment and which will lead to increased survival rates and quality of life, obviously. But there's also a big challenge to get better tests for cancer drugs. We know that there's a huge amount of money spent on drugs, so-called precision drugs, that are actually not precise at all because their relevant companion diagnostics are lacking. Next slide, please. Nowadays, focus in advanced research groups is on predicting therapy from DNA analysis by whole genome sequencing, but that is certainly not enough because it cannot capture the whole biology of cancer, which also is dependent on accessory cells in the tumor microenvironment. So the phenotype is caused by this 1% of DNA that is actively transcribed and translated to proteins. Next slide, please. So at Predict Diagnostics, we are developing CERN-seq tests. That's a method of high-throughput targeted sequencing of RNA insertion probes. And actually what we do is we detect malignant biological pathways that are actionable with the available precision medicines. And because these pathways are dependent on multiple gene activities, of course, we apply artificial intelligence, machine learning, if you will, to develop algorithms that predict disease or response to treatment. Next slide, please. So our focus is on lung cancer because we have some proof of concept, which I will show later. And it is, of course, a cancer type to which everything what I discussed before apply. So huge amounts of overtreatment. May I have the next slide, please? So what we have done is we did a proof of concept study with this technique with high throughput sequencing and high multiplexed sequencing of some 700 gene transcripts in some 100 lung cancers. And what you see here is that in only 22 of these cancers, specific targetable tyrosine kinases can be seen, like ELK, MET, or EGFR. And that explains probably why many clinical phase 3 studies fail and do not reach clinical endpoints, because there's this heterogeneity in the tumors. Next slide, please. In another 15% of cancers, we find overexpression of immune checkpoint proteins, and it will be obvious to you that if these proteins are lacking, then application of immune checkpoint inhibitors would be quite futile. And probably this explains also the huge rates of overtreatment of cancer patients with these therapies. In fact, these patients may die from the therapy instead of from their disease. Next slide, please. So we have biomarker expertise. We can look at mutations in active genes that are involved in lung cancer specifically. And also look at cells in the tumor microenvironment, like immune cells, etc. And in fact, we can now detect 1,200 gene transcripts in one test, and again in high throughput. And we have close collaboration, are connected to academic hospitals. Next slide, please. So we are very eager to join an IHI consortium with partners in the field of technology that can provide us with image-based biopsies and predicted biopsies, lung lavage, that sort of stuff. Pharmaceutical partners who wish to extend their portfolio with successful drugs by stratification of patients to be recruited to clinical trials, and of course, clinical institutes that are carrying out clinical studies. Next slide, please. So my name is William Leenders from Predica, and I present this on behalf of my partner, Marco de Boer. Thank you.
Thank you very much, William, for your presentation. Now the next speaker is Tina.
Good afternoon, everyone. I am Tina Smets from VITO, the Flemish Institute for Technological Research in Belgium. And we are looking for a consortium in the context of protein biomarker validation in neurodegenerative and other diseases. Next slide, please. We can contribute as a partner to consortia in need of analytical protein biomarker validation based on mass spectrometry, LC mass spectrometry. And we can also provide expertise on high throughput proteomic analysis using the Olink platform technology for targeted proteomics. Moreover, we can also contribute to consortia seeking extracellular vesicle expertise. Next slide, please. In addition, we can also offer access to our LCMS-based datasets resulting from biomarker discovery studies in CSF and CSF-derived EV research concerning the stratification of Alzheimer's disease, Lewy bodies dementia, Parkinson's disease, including Parkinson's disease, mild cognitive impairment, and PDD. Moreover, we can help out with mass spec expertise in large clinical cohorts at the single cell level, and we're also certified as an Olink assay provider. We have significant expertise in extracellular vesicle isolation and characterization in a variety of sample types, including brain-derived EVs from plasma. Next slide, please. We would be very eager to contribute to consortia with our expertise given analytical validation. So anything going from research institute having access to large biobanks, regulatory specialists, patient organizations, SME biotech and pharma companies. So if we can support you in your research, please reach out.
Thank you very much, Tina, for your presentation. Now the floor is to Aurélie.
Thank you. My name is Aureli SORIA-FRISCH. I come from the company Starlab Barcelona. Next slide, please. We would like to apply to topic 3. Since we think that brain health requires a new approach, so we are looking for opportunities to join a consortium in neurological and mental health diseases. As you might know, so the development of drugs for CNS is a high-risk business, and we think this is due to the lack of modern decision tools that impact the misdiagnosis rates, the large misdiagnosis rates, and the fact that we are often dealing with very heterogeneous clinical populations in the clinical trials. Next slide, please. So our solution offers neurotechnology as a service. So this is an easy-to-use EEG device, also with options for electrical stimulation in case the treatment is also sought. That can be used also with dry electrodes and semi-dry electrodes as the ones that are shown in the slide, and that it's CE and FDA certified for conducting clinical trials. Next slide. So on top of this, we have built a machine learning platform for over the 10 last years with some proprietary data sets and a proprietary framework that allows us to have a reliable EEG analysis in different diseases. So we provide for our clients protocol preparation and design. So we collect the data with a GDPR compliant platform, and we can apply machine learning for brain health, trial recruitment, and drug response in different setups. Next slide, please. This is one of our success cases in the past. So with data set of 750 Parkinson's disease patients. We have shown that we are able to find out biomarkers that can be used in the Parkinson's disease early detection with a sensitivity of 83% and a specificity of 99%. We think this can be translated into a very simple test of 15 minutes, where you can acquire data that is automatically uploaded to our cloud AI engine, and that allows you to select 60% of the patients for your clinical trial that are at the earliest stage of disease and where it is expected that the drug treatment will work best. Next slide, please. We have not only experience in neurodegenerative diseases, but we are also participating in a clinical trial, for instance, of Down syndrome, where we are basically using the EEG technology I have shown for measuring drug response to different dosages. We are also active in Alzheimer's disease and minimal cognitive impairment, and we have some biomarkers of emotional response that can be very useful in depression clinical trials. We have also participated in some IMI projects in autism and other neurodevelopmental disorders that we will also be happy to contribute to any building consortium that would like to add our expertise. And thank you for your attention.
Thank you very much, Aureli, for your presentation. And the next speaker now is Jean-Philippe.
Welcome to my pitch. My name is Jean-Philippe THEURILLAT, and I'm running a lab in the Institute of Oncology Research in southern Switzerland. My pitch is related to profiling prostate cancer progression for biomarker development. Next slide, please. Prostate cancer is a lethal disease, and as all cancers, the cancer progresses from primary to early metastatic and late-stage metastatic disease. And so there's a need for biomarker and predictive biomarkers that predict the response to surgical treatment, but also medical and pharmaceutical treatment later on. And so to have a better understanding for disease progression, to develop better biomarkers, we need a harmonized large data set of biological relevant features. And RNA sequencing data provides the relevant output of the genome. Next slide, please. However, to make sense of RNA sequencing data sets when merged together, it's really important to consider the batch effects that are coming from merging process data, even after normalization and batch correction on the left. And so you can see in many cases, even if you merge the data sets, the data set differences are much greater than the biological variance within one data set. So we have overcome this major hurdle in this field, and by reprocessment of raw data using a specific pipeline. And so you can see that now on the right, and the pink dots emerging from different primary prostate cancer cohorts overlap with another. And so next slide, please. That enabled us on the left to produce the first large harmonized data collection for RNA sequencing data related to prostate cancer with more than 1,200 clinical samples. And using that data set, we could use a trajectory inference analysis to model and quantify disease progression using a pseudo-time progression score. Next slide, please. This enabled us in turn to produce or to validate that pseudotime is an independent prognostic marker in early stage, but also late stage disease. Next, please. And we have over the years invested a large amount of effort to now basically produce a professional web tool where you can visualize, but also analyze this data set. Next, please. And so the vision is now to produce a pipeline where you can upload your own data and to visualize that own data together with atlas data of a specific disease type like prostate cancer, and then do sub-analysis to develop biomarker and validate your drug targets. And on the long run, we'd like to develop a medical device which upon the fingerprint, the transcriptional fingerprint of your patient, you can infer drug responses based on retrospective data. Next, please. And so in summary, we provide a solution for harmonizing RNA sequencing data so that they can become interoperable to mine and exploit disease progression for biomarker development, an interactive web tool to visualize and analyze harmonized RNA-seq data together with external data. And if you're interested in my expertise, please contact me through the portal, the platform, or through email, and I'm happy to join your team. Thank you very much for your attention.
Thank you very much, Jean-Philippe, for your pitch. Now our next speaker is Jérémie.
Good afternoon. I am Jérémie THIBLET from Neoventure Biotechnology Europe, and I will present the PAMD project in collaboration with the eye clinic of University of Thessaly. Neovascular age-related macular degeneration, also known as wet AMD, is a prevalent progressive retinal degenerative disease that causes gradual vision impairment. Our project aims to validate biomarkers that can guide the selection of the optimal treatments for individuals with NAMD. Next slide, please. Okay, it's good. NAMD poses a significant challenge due to the diverse responses that patients exhibit to anti-VEGF therapies, the primary treatment method, leading to concerns about the sustained efficacy of these agents and the optimal choice for each patient. Extensive multivariate analyses are therefore required to predict response to anti-VEGF treatments and support treatment decisions. Our goal with this project consists of implementing a novel methodology to progress the clinical use of plasma biomarkers to predict response to treatments, relying on established correlation with advanced imaging technologies and clinical variables. It will lead to robust clinical evidence supporting a solution ready for regulatory acceptance and the development of qPCR-based molecular diagnostics for predicting treatment response to anti-VEGF agents. Next slide, please. Through this project, we will implement a new method using plasma biomarker-based approaches for clinical assessment of patients to support medical management decisions regarding agents to be selected and the associated treatment protocols. The project will validate an accessible and affordable biomarker testing technology, contributing to improved patient outcomes, reduced therapeutic burden, and decreased economic strain on the European healthcare system. Next slide, please. The project includes a comprehensive biomarker validation plan. We will employ an innovative DNA aptamer technology in correlation with the latest imaging technologies to validate biomarker patterns and develop a qPCR test measuring corresponding epitope signatures in plasma. Our activities will also include the establishment of a digital collaborative sharing platform for data analysis, multi-center validation studies, collaboration with patient advocacy groups, and the development of a robust regulatory and market acceptance strategy. Next slide, please. We bring expertise in biomarker validation and testing using DNA aptamer technology enhanced by machine learning to create qPCR-based molecular diagnostics. We also bring high-level medical and imaging expertise related to NAMD. Next slide, please. Therefore, we are looking to collaborate with pharmaceutical partners, developing treatments, clinical researchers, and healthcare professionals with expertise in NAMD. In conclusion, this project represents a pioneering effort to transform the management of NAMD by applying an innovative methodology for validating complex disease biomarkers and making them accessible for clinical use. We invite you to contact us and join us in this project. Thank you.
Thank you very much, Jérémie, for your presentation. And now the next speaker is Aad.
Yeah, thanks. Aad van der Lugt is my name. I'm representing the European Imaging Biomarker Alliance, which is part of the European Society of Radiology. The European Society of Radiology is a professional society in the clinical imaging domain with more than 50,000 members. So making use of our expertise gives you access to a lot of clinicians and imaging experts for implementation of the results of your consortium. Next slide, please. Imaging biomarkers can be considered as part of this goal, as these biomarkers are relevant for diagnosis, monitoring disease progression, selecting the optimal therapeutic treatments, or assessing treatment responses. You see some examples demonstrating a brain tumor, an increase in size of a lung nodule, which is actually lung cancer. The second one is response to treatment with a decrease in size of lesions, and also the last one, nuclear medicine exam, demonstrating the effect of treatment. So with imaging, you will get information on the structure and functional information from normal or diseased parts of the human body. There's of course a problem related to imaging biomarkers. There's a huge heterogeneity in acquisition and quantitative analysis, and there's no real consensus in the methodology for technical and clinical validation, and rather frequently clinical validation is lacking. So what we try to achieve is the development of so-called imaging biomarker profiles, which is a protocol for acquisition and analysis, which include one or more performance claims. For example, it tells you what can be achieved by following the profile. For example, the accuracy of an imaging biomarker, coefficient of variation of percentage change that can be measured. And that is actually based on the expertise and methodology developed by KIBA, which is currently the international standard. The European Imaging Biomarker Alliance has collaborated with KIBA to develop this type of methodology. Next slide, please. So what can we do and what is our plan? We can promote and teach the right methodology for technical imaging biomarker validation based on rigor methodology rules. We support the imaging community with clinical imaging biomarker validation, and also very important, we support the imaging department's implementation of these biomarkers in clinical practice, which is of course relevant in case you reach the end of your project and you would like to promote the use of your biomarker in the clinical domain. Next slide, please. What can we offer? Expertise in technical validation. So we can organize repeatability studies, reproducibility studies to assess inter-scan variability, but also inter-observer variability and the variability between software packages. We have a large network of centers that has an expertise in technical biomarker validation that can be used for these activities. We can provide access to large data sets for validation of image analysis algorithms, which is relevant for defining the type of acquisition needed for optimized image analysis, especially automated image analysis. And then we have the expertise in deploying the right methodology for the technical biomarker validation. Once the profile has been defined, you can use the results for your clinical validation. We have expertise in that domain. We can give you access here to a network of centers of excellence, and these are selected based on an excellent collaboration between imaging and clinical departments. And finally, we can provide expertise with the implementation of biomarkers, as we have a network of professional societies like the European Society of Radiology, but also other imaging societies. And of course, project management can also be offered by our community. Last one. So my main message is please include us in your proposal and in your consortium, as we have expertise that is currently lacking in most of the projects, especially with regard to the optimal way of validating imaging biomarkers. Thank you for your attention.
Thank you very much, Aad, for your presentation. Now we will move to the last pitch, and John, the floor is yours.
Okay, so I'm also interested in imaging biomarker validation. And the next slide. So our perspective is that there are really two sorts of biomarkers. The biomarkers where you take something out of the body from blood tests and biopsies, and the biomarkers where you don't take anything out of the body, but you just receive a signal, such as imaging or electrophysiology. And these second ones, as we've heard quite a bit this afternoon, are ideal for public-private partnerships. They have different IP landscape, but more importantly, they have a very different validation roadmap. As has been already said this afternoon, that standardization and harmonization is a hard problem. And also from the point of view of the innovator, a lot of academic innovation fails to translate, or it translates very slowly or not at all. And that's a missed opportunity for using the outcomes of academic innovation to drive healthcare. Myself and Bioxidine have co-led a couple of previous IMI IHA projects in imaging biomarker validation, and have put an imaging biomarker into the FDA's biomarker qualification program. And we would be looking to collaborate to work in important disease areas to further the validation of imaging biomarkers, including evidence of either repeatability or reproducibility, which depends on the context of use. They're not equally important in all contexts of use. Guidance, metrology, particularly metrology for hardware, software, phantoms, workflows. And we're interested in really all imaging, particularly MR biomarkers, both existing that need to be validated in new contexts of use, and new and innovative ones arising out of academia. Next slide. So we would divide this into 4 pillars of activity. Repeatability, same subject, same scanner, particularly important in pharma trials where you're looking at response, pharmacodynamic, monitoring biomarkers. Reproducibility, looking at the comparability across platforms and across centers, is much more important if we're interested in companion diagnostics, safety biomarkers, prognostics, predictives, and so on. Effect size is important, particularly in response biomarkers, and the interpretation, particularly interpretation of negative results, is really important for the drug developer, and that needs careful design. Our contribution includes designing protocols, implementing, acquiring, analyzing, reporting, and the metrology that underpins that. Next slide, please. So we have extensive expertise both in pharma trials, academic studies, across all therapeutic areas, with a fully regulatory compliant platform. The next slide. We're very interested in looking for consortia involving large companies or academia in any therapy area. We're interested in collaborating in regulatory science, metrology, and statistics with innovative phantoms. The next slide just gives an illustration of the roadmap for, in this case, for oncology, that we have developed under previous IMI funding, that is very different from molecular biomarkers. Thank you.
Thank you very much, John, for your presentation. So this was the last presentation of today for topic 3. As a reminder, we will publish the presentation just after the end of this session, and also the recording will be available on our website and on the B2Match platform. Also, if you are interested in any of the presentation, please reach out to the pitchers directly on the B2Match platform. So either you send them a meeting request or you can message them as well on the platform. I would like to thank you all the speakers for their presentation and respecting the time that they had, 4 minutes. It goes quite fast. And I would also like to thank everyone who attended today's session. We can officially close the IHI call days for call 6 and 7. This was the last session for the IHI call days. I would like to thank you again all and wish you a very nice day. Thank you. Bye-bye.